Okay, folks. Today we will discuss DNA cloning in molecular biology. So I'd like to divide this lecture into following sections that what is source of DNA for cloning, or what vectors we have been using for cloning, source of DNA that could be either it is genomic DNA that, for example, you have some gene and you want to clone it, either it could be genomic DNA, it could be cDNA, it could be like any, for example, gene fragment which you want or promoter, whatever you want to clone, that could be the source of DNA vector. For example, we have been using different vectors. For example, plasmid vectors, which have been used most frequently, and along with that one, there could be cosmates or yeast artificial chromosomes or bacterial artificial chromosomes for larger DNA fragments that are used as vectors. Restriction enzymes that are conventional type 2 restriction enzymes, which we have been using for restriction enzyme based cloning. However, today we have other methods of cloning as well. For example, Golden Gate cloning or gateway cloning method. Uh, in gateway cloning method, we use, for example, combination sites for example, LR clonase we use and BP clonase, so LR sites and BP sites. Similarly, in Golden Gate cloning, we use only one restriction enzyme like BSA1, and then we use, for example, just one restriction enzyme for cloning multiple DNA fragments. And that is also directional cloning as well, that we can keep the directionality of all those 10 fragments if we want to clone, for example. So that will be cloned in a particular sequence, and that there will be only one enzyme that will be used, for example, BS1. And then ligation, that we use DNA ligase to ligate the like insert and the vector. Then bacterial host for, because mostly we use plasmids and bacterial plasmids and like E. coli uh, for cloning DNA or a gene, for example. And then bacterial host, we will see, for example, we can use, for example, chemical competent cells or like uh, electrocompetent cells we can use for um bacterial transformation as well. So transformation like chemical competent cells that could be used for heat shock, or electrocompetent cells that could be used for electroporation. And then we will also see that, for example, if you, for example, want to transform plants or animals, then you can use different transformation methods like ballistic transformation, or, for example, um, like in animals, for insects, for example, we use transfection and what you call that, microinjection as well. Selection of recombinants, we will also see that how we select those recombinants in, for example, bacteria. So we use LAGZ gene. So what LAGZ encodes for, that encodes for beta galactosidase. Beta galactosidase hydrolyze the lactose into glucose plus galactose, but it also hydrolyze xgal. So that will be hydrolyzing xgal into glucose plus a blue pigment, blue colored compound. So that will give you color visible selection of the transformants. So what we do in selection of recombinants, that we divide the LAGZ gene into uh, two parts, that is alpha domain, that is alpha part, and the beta part. So in alpha, for example, one of those, that is alpha, we have multiple cloning sites that are present where we can insert our, for example, foreign gene. So if that part of foreign gene that will be inserted in alpha component, then what would happen that the both part that would not generate an active uh, beta galactosidase. So what would happen that would result in that would result in white colony. However, if that would result in active beta galactosidase, that would give you um, blue colonies. So whenever, for example, blue colonies, that would mean that your insert that has not been cloned in your vector, like plasmid. However, if there are white colonies, that means that your insert has been cloned in uh, your vector. So what cloning means? Cloning means that to produce identical copies, that either it could be of a particular gene or a DNA fragment, uh, or it could be to produce identical copies of a cell. So clone means that identical copy, and cloning means that to produce identical copies of a particular gene or a particular cell. Then for that purpose, we use recombinant DNA technology, that we manipulate genes, and then we use different like tools for that one, in which we just, for example, saw in the previous slide, that we use vectors, and then we use bacterial hosts, and then using those bacterial hosts, we produce identical copies of that particular gene. So here is, for example, we identify a particular gene, and then, for example, we isolate that gene, and then we insert that gene into bacterial plasmid, and then we transform that bacterial plasmid into bacterial host, and then that plasmid that will replicate independently of bacterial genome. So that will produce multiple copies of that gene, and then we can, for example, use that recombinant plasmid for our own beneficial purposes, for example, transformation of plants, or, for example, for medical purposes, that expression of gene of interest to produce protein, and we can use it for medical purposes as well as applied purposes, as well as basic science research. So, clone genes, most of the methods for cloning a piece of DNA in laboratory share general features such as bacteria and their plasmids. So, what are plasmids? Plasmids are small circular DNA molecules which can replicate independently. That their replication does not depend on replication of bacterial genome. They can replicate independently, and then they would have particular genes, for example, for genes for antibiotic resistance that would be present on plasmids as well, and then those plasmids would have independent replication origin as well. Then those plasmids would have, for example, low copy number as well as high copy number plasmids are there as well. Then gene cloning, that a foreign DNA that would be inserted in plasmid, and then recombinant plasmid that would be inserted into particular bacterial host cell. Then those cells, in those cells, the plasmid that would replicate, and then it will produce the multiple copies of that uh, insert. So why genetic engineering? So genetic engineering have been used, for example, all those these methods, for example, identification of gene, and then isolation of that gene, and its insertion into particular bacterial plasmid, and then transferring that plasmid into bacterial host, and then using that bacterial host to transfer that, for example, particular gene to either plants or animals. Uh, all these methods include, for example, genetic engineering. Today, for example, there are even genome editing as well. So Genetic engineering could be used for medical purposes as well as health applications. Similarly, it could be used for production of novel and important proteins. For example, insulin that could be produced, the recombinant insulin you might have heard that could be produced using genetically engineered methods. So, for example, here the recombinant insulin gene that could be separated and then transformed into plasmid, and that plasmid that could be recombinant, particular insulin gene that could be isolated and then inserted into plasmid, and then that plasmid that would be inserted into, transformed into bacterial host, and then it will produce the recombinant insulin protein. Similarly, genetic engineering that have been used for agriculture applications, for example, GM crops. Pakistan, for example, have been growing BT cotton, that is GMO, that have been produced using genetic engineering methods. Similarly, GM maize, and then we also have, for example, GM soybean. So all those are produced, for example, for different beneficial purposes using genetically engineered techniques, genetic engineering techniques. Cloning genes for scientific studies along with medical as well as health purposes, and then agriculture purposes. The cloning of genes, that is also, the scientists have been using these techniques and cloning of particular genes for basic scientific purposes. That's doing research as well, functionally characterizing those genes, for example. So let's see the basic of cloning. That what you need for example, you need a source of DNA that could be the genomic DNA, that could be either it could be the cDNA, uh, or it could be synthesized DNA. Then choice of vector, as we said, that choice of vector could be plasmid vector, could be cosmid vector, could be yeast artificial chromosome, could be bacterial artificial chromosome. Restriction enzymes you have been using, for example, from 1980s. So there are type 2 restriction enzymes, for example, which recognize particular sequence, and their natural function is to defend the bacteria against invading viruses. So these restriction enzymes, for example, they have been used. There are two kinds. For example, the blunt end restriction enzymes, which produce the blunt ends when they cleave the DNA, they produce the blunt ends, and then there are some of those which produce the sticky ends. So we can also use two enzymes for keeping directionality of cloning, and then DNA ligase to ligate the vector, to ligate the insert into vector that results into recombinant DNA or chimeric DNA. Then host cells, that there are different, for example, bacterial strains. We have been using DH5 alpha or top 10, etc., which we have been using, and different transformation methods we have been using as well. For example, chemically competent cells, in which we have been using calcium chloride, while electrocompetent cells for electroporation, in which we use electroporation for transformation. So source of DNA, as we said, that either it could be genomic DNA, it could be cDNA, it could be amplified DNA, or it could be synthetic DNA. Then vectors, in which we can ligate the foreign DNA and foreign gene, uh, and that carries the foreign DNA, that or foreign gene, into a particular host, bacterial host. For example, and then maintain the foreign DNA in the host cell, then replicate and then pass to the next cell during cell division. Um, so there are plasmid vectors bacteriophage, lambda, M13, cosmates, phagmates, bacterial artificial chromosomes, yeast artificial chromosomes. So mostly we use plasmids, but sorry, but the size of plasmids is limited. That their capacity to carry the foreign DNA that is limited. This one is, for example, uh, plasmid DNA. So here, those multiple cloning sites they have been indicated, in which you can use these multiple cloning sites to insert your the specific gene or particular gene into these sites, and then that plasmid vector that will be transformed into particular host for replication and producing its identical copies. Those plasmids that could be high copy number or low copy number as well. So one of the plasmids that is PBR322, a high copy number plasmids, in which you can see, for example, this one is uh, different enzymes which have been indicated on that one that are present, and then tetracycline tetracycline resistant gene that have been indicated here. Multiple cloning sites would also be present, in which you can use those multiple cloning sites, and then you can uh, copy, you can clone that particular gene into electronic site. Type 2 restriction enzymes, as we discussed, that those recognize a particular sequence and then cleave on that particular sequence. That could be blunt end producing or sticky end producing enzymes. Uh, some of those restriction enzymes they recognize six nucleotides, while six base pair sequence, while the others recognize four base pair sequence. Here, for example, the sequence of ND3, all those sequences are palindromic. Palindromic sequences means that if you read from the forward or reverse direction, that would be same. So AAG, CTT, that ND3 recognize AAG, CTT, ECOR1 that recognize GAA, TTC. So these are the restriction in enzyme PST1, FAT1, SSP1. So these recognizes these sequences and then produce the sticky ends. Sticky ends and blunt ends. Blunt ends that there will be no overhang, while in sticky ends there will be single strand overhang that nucleotides. For example, here you can see that there will be TTAA overhang uh, in sticky ends. So those overhangs that could be particularly useful for cloning purpose. In this one is also same, that restriction enzyme ECOR1 has been shown that when it cleaves, it produces the sticky ends which have the overhangs, and those overhangs that could be used for cloning of foreign DNA. So here you can see that the particular insert that could be inserted in plasmids using those overhangs. Then bacterial transformation, as we discussed earlier, that could be either with chemical competent cells that could be produced with calcium chloride, or that could be produced with uh, electrocompetent cells, and then electroporation that could be used for the transformation efficiency of chemical competent cells that is lower than electrocompetent cells. The efficiency of the electrocompetent cells that is much higher. What it do, that calcium chloride treatment that produces the that small that pores, and that results into that the bacterial cell wall that becomes permeable, and then the foreign plasmid that could be introduced into bacterial cell. Uh, Selection of transformants that, as we discussed, that we use, for example, antibiotic resistance genes for selection of those plasmids. Directional cloning that you can use two enzymes for the directional cloning of your particular gene into plasmids. Dephosphorylation, that we also use dephosphorylation, that we use phosphatase enzyme to dephosphorylate the phosphate group, phosphate group, and that would not allow to self-ligate the plasmid. So dephosphorylation will uh, like inhibit the auto ligation, that self-ligation of the plasmid DNA. If we dephosphorylate, that would inhibit the self-ligation. Then blue-white selection, which we use, in which we use the LAC-Z gene, that is responsible for coding beta galactosidase enzyme. Now, what does beta galactosidase do? It converts the lactose into galactose and glucose. So we can break down the LAC-Z gene into two parts, alpha and beta, each coding for a fragment for beta galactosidase enzyme. So LAC-Z alpha and LAC-Z beta, and when it will be become an active form, that would convert the lactose into uh, glucose and galactose. But it can also break down the hexagon into a blue pigment and glucose. So when we transform the plasmid into bacterial host, then we, for example, use when we clone our particular insert into multiple cloning site, for example. So those multiple cloning sites are present in LAC-Z. LAC-Z gene. So when your particular insert that would be cloned inside those multiple cloning sites, the LAC-Z gene that will be disrupted, and ultimately that would not be able to produce active beta galactosidase. So when active beta galactosidase that would not be produced, that would result in white colony. However, when your insert would not be present here, then multiple cloning site would remain as such, and uh, beta galactosidase, active form of beta galactosidase, that will be produced, and that would result in blue glutamine. So using that one, uh, the LAC-Z gene, we use the blue-white selection colonies that. So here, for example, white colonies you have been watching with insert, while blue colonies without insert. So these are the summary slide, that basic steps, that you have a particular insert, then you can clone it into vector, and then you can transform that vector into particular host and bacterial host, and then it will replicate inside bacterial host to produce identical copies. That is called as cloning. That's all about your cloning lecture. If you would have any questions, you can ask. Feel free to ask. Thank you.